Hello, everyone. I'm Khadija, a digital editor, and thank you for attending today's webinar, The ABCs of ADCs from PK to PD. Before I introduce you to our host and speakers, I'd like to quickly cover a few housekeeping items. Firstly, all the information about today's webinar can be found on the right-hand side of your screen, including the Q&A tab. You will have the opportunity to submit questions to today's presenters by typing your questions into the Q&A box on the side of the window. You can send in your questions at any time during the presentation, and you will find all of your submitted questions in that tab. Please note that other attendees will not be able to see your questions. We will collect everyone's questions throughout the webinar and address them towards the end of today's sessions. If you encounter any technical problems during the webinar, please use the online chat support function, which can be found by clicking on your profile icon, navigating to the help desk, and clicking on the chat with support icon. Alternatively, you can contact us at clientprojects@tnf.co.uk for further support. An on-demand version of today's webinar will be available shortly after the event. Thank you again for joining us today. I'll now hand over to David Good, who will be hosting today's event.
Good morning, good afternoon, and good evening, everyone. Thank you so much for joining us today, where we will be discussing the ABCs of ADCs from PK to PD. In this webinar, we're going to be talking about predictive bioanalytical strategies for the development of antibody-drug and antibody-oligonucleotide conjugates. Before we get into the meat of the webinar, I do want to quickly discuss a little bit about Illyri and who we are. Some of you may still not be familiar with our name, but we do have over 30 years of experience. We are essentially three independent labs who have come together. We used to be Pixent Labs, EMA Biotech, and Tandem Labs. So again, we have more than 30 years of experience providing bioanalytical CRO services, and we are a global company with 2 sites within the United States and 1 site in Europe. Oh. And I am hung up. Let's see. All righty. So today we want to talk about ADC development. As many of you know, and why you're probably here, there has been an explosion of interest in antibody-drug conjugates with more than 400 ADC programs globally currently, and you can see the increase over the past 2 decades of those programs. What we are seeing, interestingly, is that in addition to the initial primary indication of oncology, there's been a rapid expansion into new indications where ADCs could be used. Unfortunately, while ADCs do have very high potential, they're extremely complex, and we still see a very high attrition rate, a very high failure rate of these compounds. And what we see is that most of these failures have stemmed from unmeasured biology, be it the target not being feasible for the indication, drug not reaching the tissue or the site of action, or off-target payload accumulation in normal healthy organs, a mechanism perhaps not fully being engaged, or the inability to appropriately predict toxicity. So today we're going to be discussing what are those questions that you should ask when developing one of these therapeutic compounds or an ADC. Elucidation, is your target really appropriate for the indication that you're trying to hit? Exposure, how much antibody and payload reach circulation or reach the actual site of action? Delivery, is the ADC engaging the correct target cells in situ? Mechanism of action, right? So is the expected biological effect being achieved? And prediction, can we anticipate the efficacy of the compound and the safety outcomes for your drug? So it's my privilege to introduce you to 2 of our subject matter experts in this area who will be leading this discussion today. Dr. Corinne Ramos has more than 25 years of experience in BioA, especially on the clinical assay and biomarker development front. She's uniquely positioned to work with many of you, as she's one of the few people that I know that has worked in all aspects of scientific development, be it government labs, academia, large pharma, and the CRO space. We also have with us our Senior Laboratory Director, Dr. Troy Voelker. Troy has more than 20 years of experience within the BioA space. And not only is he a member of AAPS, but he's actually the chair of the oligonucleotide discussion group. Troy is extremely well known within the industry, and we're lucky to have both of these speakers leading this presentation today. And with that, I will hand it over to Dr. Corinne Ramos.
Thank you very much, David. So ADC performance is determined by the three inseparable layers that you are seeing in that particular slide. What exposure do we achieve? Where does the drug go? And where it's there, what does it do? So if any of those layers fail during, the development, the ADC, program is really at risk. And this is the structure of our talk today: exposure, delivery, and biological effect, and how integrating these three different layers can give drug developer the real mechanistic confidence. But first, a little bit, on the ADC structure. Each ADC structure component introduces its own variable. Could be stability, potency, diffusion, toxicity, specificity. Our work, in bioanalysis, is to be able to quantify how each part behaves in a biological system in order we can anticipate efficacy and safety. But first, before engineering the ADC, one critical question is to understand if the potential ADC target is viable. So a viable ADC target must be highly expressed on disease-driving cells, low or practically absent in essential normal tissue, and has to be as well surface accessible that allow the antibody engagement. And this is where spatial capability and spatial analysis is really shining. Because in contrast with bulk analysis, where you can measure the expression of any antigen in a tissue lysate, the spatial capability will help you to really nail down the heterogeneity of distribution of this target on the tissue sample, will tell you exactly what niche or what environment that are going to be a little bit preoccupant for ADC target. So this type of experiment needs to be done in advance, of ADC engineering, because it will prevent advancing undruggable target, for example. And we also need to measure if, the antibody will be able to reach that target. And spatial, again, capability and evaluation, on tissue is the way to go, because it will help you to understand if that target is in a very defined zone that are not accessible, to ADCs. For example, in that particular example that I have on that slide, you can have, two different targets of interest, but one can be trapped, in a very dense, stroma, or in a region that it's also, not very accessible, for the ADC. But the target B, for example, it's in completely in an open zone and can be totally accessible, to an ADC. And that has consequence in the way as well you can support rational selection of the payload and as well the linker for your ADC. Because if you have, a target that is very not very accessible or very difficulty of being reached, you can think about developing, a more cleavable linker or more, a diffuse payload. And in contrast, if you have an open, target, you can think, a much more controlled system. So to summarize, before engineering, we must confirm which cell type express the target, if the target is membrane localized, what is, the spatial distribution of this target? Do we have that target in express as well in normal tissue versus, the diseased tissue that it's intended to be targeted? And there is as well, some co-expression pattern that could be relevant for really selectivity of the target as well as off risk. And this altogether really confirm, through, for example, the target scoring, comparison or target scoring system that I put it on that slide, can confirm really the real target druggability in tissue. And it can be a very early go, no-go decision in terms, of developing and designing the right ADC. I'm going to hand the talk to my colleague Troy now for exposure.
Thank you, Corinne. Obviously, after you have determined that you have a viable target and the best way to get to that target, you are then going to need to measure the, concentrations of both systemic and, what is reaching those tissues. So ADCs, obviously, they take up a lot of, they've got a lot that are involved in them. There's an antibody, there's a linker, there could be a small molecule, there could be an antisense type of payload, and then the drug itself may have the limit of one drug on it, or it could be up to potentially even 8 drugs on that, and that's what we call the drug-antibody ratio. So to thoroughly characterize the ADC pharmacokinetics, regulatory agencies do require measurements of multiple parts of these analytes. So what is the strategy that you need to take with multi-analyte measurements? We got to do the total antibody, so a measurement of all of the antibody species, regardless if there's a payload that's attached to it. It reflects antibody clearance and its disposition. Typically, this is done by LBA, but a lot is being done by immunocapture, mass spec. You also need to measure the free payload, so that's the unconjugated drug that is circulating. It's a measurement that's, a release of the small molecule or the antisense strand. It's critical for the safety because that represents the potential toxicity of those molecules. And the reason a lot of these ADCs are being produced is because these molecules are very toxic. The idea is to do a dose that is targeted rather than typical chemotherapy where you're just doing a full dose of these small molecules. This traditionally and still remains to be an LC-MS-based type assay. Then you have the conjugated payloads, the payload that is still associated to that antibody, it's still on the linker. You got to get a difference between, is there some type of linker stability that you need? What is the amount of deconjugation? Once again, this is usually an immunocapture LC-MS-based type strategy in order to get that measurement. And then you have the total drug and total payload. So a measurement of both total amount of the payload and the total amount of the antibody. This can reflect linker cleavage, a systemic type payload exposure. This can be done via LBA, but a lot of it, most of the time, is done by LC-MS. So other requirements that are involved in ADC drug development is anti-drug antibody evaluation. So these are antibodies that are produced within the system that can target the drug itself and limit the exposure. So it can alter the PK, it can reduce the efficacy, you can get into some safety issues due to you thinking that the drug is supposed to be doing something and it's doing something else. It can interfere with the target binding, and it can affect the DAR, or the deconjugation and the payload stability. So it also can interfere with some of these assays, whether that be immunocapture or LBA. And then you have the DAR assessment. So that's the assessment of the average number of drugs that are associated to each antibody. Now, DAR itself can affect precision of an assay. It also can affect the efficacy if the DAR is expected to be higher, but yet it's not there. So it can affect the PK/PD behavior. So the manufacturing consistency is very important, and it is a part of FDA requirements to know what that measurement is. So ADC in biological matrices, what is that regulatory expectation? Obviously, for preclinical, you're required to do TK and toxicology. That usually is done in either plasma or serum, depending on whether the LBA assay is using serum. Usually LCMS is using plasma. And you may have to do some whole blood assessments, whether the linker is stable or not within the whole blood environment versus the plasma and serum environment. Obviously, there's tissue measurements, whether the target is liver, kidney, spleen, or some type of tumor. Most of that still done via LCMS, still can be done LBA. And then there's other optional risk-based measurements. A lot of these targeted payloads do have ocular accumulation. So there may be some ocular tissue studies, CSF, urinal bile, depending on what type of elimination pathway you're looking at. Then when you get into clinical assays, once again, looking at primary PK, and then the total antibody, conjugated ADC, free payload, and then metabolites that are associated to it. Whole blood measurements may still be required. It obviously depends on the red blood cell partitioning of it, and then critical other factors such as hydrophobic payloads such as MMAE and MMAF. Okay, so why LCMS for a multi-analyte strategy? Well, I want to point out that every measurement that's done independently is going to require another aliquot. So as much as you can get done in a single measurement, the more you're going to save of that critical sample. So you're going to do the small molecule LCMS. If you can devise a scheme where you can get both the small molecule and the conjugated payload done in a single assay, you're going to save those samples. So, we'll talk about the small molecule strategy, whether it's protein precipitation, SLE, SPE, but you can also get the conjugated antibody analysis via a immunocapture. You may be able to directly do a enzymatic digestion and then follow a surrogate peptide along in order to get that measurement. And then when you're doing the total antibody, there's a lot of pathways that you can go down. That could be the, once again, a top-down type or a bottom-up with the LCMS, with the digestion, once again, following the peptide. You could do a top-down, or you can even do a middle-up. It just depends on what route is going to give you the sensitivity that you need and the specificity. So when looking at a total antibody analysis, we often talk about immunocapture. And I think many of you already know what that is, but it's worth talking about what is that scheme. So you devise an antibody where you can do whether it's an IgG or an anti-id type of capture. It's got a biotin on it. You pull it down into a streptavidin. That then captures your targeted antibody. And then you can go through an elution. You can get an intact ADC measurement, although those usually don't provide you the lower limit of quantitation that's needed for the assay. You can do a proteolytic digestion. Once again, you're going to follow the surrogate peptide for the total antibody, and you can potentially get the payload assessment that's associated to that conjugated warhead. The other one is a linker digestion. In that case, you would get the conjugated warhead without being able to do a measurement of the antibody. So how do we quantitate antibodies via LCMS? So one, in terms of strategy, you want to keep all sample preparation factors consistent. So your digestion, your extraction, and your cleanup, all of that has to be looked at in method development in order to achieve a robust, rugged assay. You want to look at varying that antibody concentration because obviously samples are going to come, they're going to have high and low, and you're going to need this assay to work across a range of what that antibody exposure is going to be. You're going to use calibrators. Now that could be a calibrator that comes from the actual antibody, or you could use a surrogate peptide or instead of a signature peptide approach. And then you want to focus on one peptide for quantitation. It's always nice to have others that are there for qualitative measurements, but you really only want one single peptide for the measurement itself. So, you know, you need to measure this antibody. Where do you start, right? You got prior information. What's the antibody sequence? What's the property? Do you have any type of stability information? Is the linker labile under certain conditions and stable under other conditions? You want to understand the goal of study. Not every single study is going to do every single measurement. And to know what those goals are going forward will help you design what type of aliquot you can use, what type of sensitivities it's going to be required. You know, you're going to want to know the range of the assay. And a lot of times people come in, I just want the most sensitive assay that you can produce. But if you're diluting all of your samples down into that range, you really don't have an appropriate assay. You want to rely on knowledge of previous, you know, things that you have in hand. Was there linker stability in previous studies? Has this linker been used before? Has the antibody been used before? And if the antibody's been used before, are there already known peptides that I can use for the quantitation? Obviously, you need high purity. And then once you've chosen that surrogate peptide or the signature peptide, getting a labeled internal standard is very, very helpful in order to achieve good accuracy and precision. So the antibody quantification, you know, what is that common approach? So almost all of it's typically done via bottom up. So surrogate peptide or a signature peptide type approach. In method development, you want to know what those peptide targets are and where you're going to source them in order to get good, reliable, clean signature or surrogate peptides that go through this digestion. You want to know, you know, your extraction optimization. Once you get through and look at what's the best way to unfold the protein, digest the protein to get that efficient, followed by a secondary sample cleanup. Obviously, the cleaner the sample that gets to the mass spec, less matrix effects that you're going to see. Optimization of your LCMS. So we like to do orthogonal. If there's one way that we're extracting it via SPE, always like to do a secondary type cleanup on the LC column that kind of separates in a different individual manner. Validation, right? You want to avoid the inconsistent or incomplete protein extraction. This often leads to method validation failure. So once again, back in method development, understanding the ruggedness of that digestion in order to get to that protein or that signature peptide that you're following. There may be some digestion variability when it comes to each individual lot. There may be some type of interference. So once again, going through that method development, looking at several lots to ensure that this is going to work across all of the samples that you're going to receive. And obviously you want to look at stability issues. Sample analysis, you know, once again, are you at the appropriate range? Are all the samples below LLOQ? Are most of the samples above your ULOQ? So dilution of the samples is also very important. And this is where you get into that anti-drug antibody. Are there effects that when you dilute something down gives you a different value? This obviously comes into, you know, ISR evaluation. So payload quantitation, you know, we're talking about a small molecule at this point. I think everyone is very comfortable and familiar with this, whether it's a protein precipitation or a solid phase extraction. But you do have to consider if you are digesting your sample to get that free payload off, you may end up with something that is not the payload represented as the small molecule. It could have some form of the linker. It could have a few forms. You want to consider what type of metabolites are going to be there? Do I need to measure those metabolites? And are those metabolites associated to a digestion of the linker or metabolism of the linker or metabolism of the small molecule itself? So in method validation, if you've got poor precision, is that because I've got a lot of variability within the drug antibody ratio distribution? Inconsistent linker cleavage. So are you cutting across at the same point in that linker? Poor sensitivity. So the free payload is often there in a very small concentration. And so these assays are usually trying to get to the lowest limit of quantitation as possible. That often means you're taking larger aliquots, which could lead you to other types of matrix effects. And once again, stability is always a concern. You want to ensure that you've got a stable environment. You may have to add something in order to produce that type of stability within that linker to payload type association. Sample analysis, once again, looking at the same thing. Am I at the appropriate range? Is the dilution of the samples, do I get some type of dilution effect? And always ISR evaluation. So, this was often an oncology-based type of therapeutic, but other things are being used in antibody-drug conjugates. A new thing is both siRNA and ASO type conjugates. So this is a new level of discussion and something that we're excited to take on here at Illyri, because what this does is it gives you another potential handle with regards to a type of hybridization of the assay. So if I can grab on to the RNA portion of the antibody, I may be able to get around other type of immunocapture type methodologies. And this can help me with both the free payload and the conjugated payload type of measurements that will be required for these studies. So we've talked about these bioanalytical workflows, things that would be different between an siRNA study and an ASO study is one, the siRNA is double-stranded. It does require a RISC complex, the endosomal escape, and most of the time when you do a hybrid-based association in terms of extracting out those oligonucleotides, you now have to think about the sense strand. The ASO is a little bit more simple. It's single-stranded, and I can use the antisense strand to also grab onto the antibody to get that full measurement, whereas in siRNA, you'd be releasing the antisense strand, so you may not be able to get both payload and the antibody association in a single measurement. So once again, the association of RNA now gives us a secondary look at how we can extract out these samples and do those measurements via LCMS. So just a quick comparison, a measurement of the antibody. LBA has been more of the traditional, but hybrid immunocapture LCMS is definitely on the rise. Measurement of the payload has always been and probably always will be a mass spec type measurement. Sensitivity, we're starting to get better in terms of where we can achieve in terms of the mass spec. LBA is still a bit more sensitive. The specificity is very high on the mass spec, and the DAR is really a mass spec measurement along with the metabolites that are associated with those payloads. And now I will hand it back over to Corinne.
Okay, so plasma PK tells us about the exposure, but it does not really predict, where the drug is going. So there is, a multiple factor to ADC efficacy, but one of them, it's where does the drug go, and how far, the payload diffuse. And this is where, the LCMS for tissue, analysis, the mass spectrometry imaging that I'm going actually to introduce, as well as to a certain extent, the imaging mass cytometry can complement each other because it provides, information about, tissue delivery. And this can be really important, to distinguish within those experiments, if you have, a delivery failure from, a biology failure. So the mass spectrometry imaging help us to really quantify the payload in each tissue. And so you have a picture, an image, on the right of this slide that showcases, how, the mass spectrometry imaging is working. And you have, on the left, side of the image, the histological, structure on tissue that are being revealed through, an histological staining. This is, an H&E staining. And that helps us already, to compartmentalize, this tissue structure. So you can see, that you have some stromal components, some tumor components in that particular, picture. And on the right side, you have, now the molecular imaging that could represent, for example, a payload accumulation within that particular tissue structure. And you can see right away what the value of the mass spectrometry imaging here, because it helps you, like to characterize the accumulation of the payload within those different, like structure. So it's really show you where the payload is going to be delivered directly onto the tissue. And sometimes, like in the right compartment, and sometimes, like it could be delivered in a compartment that the ADC was not intended actually, like to be delivered to. In addition to it, you can go a little bit further and trying actually to understand the compound, like specific delivery by overlaying, in addition to the payload, like MSC image, something, that can characterize, like this time the different, like cell type and phenotype, like within the tissue structure. And if we can use, like a special, like proteomic platform. So by overlaying, like the payload, like MSC to the particular imaging mass cytometry image, you can really start looking at which cell, like get, like targeted, which cells, like will be the target actually, like of your ADC. And you can really net down, like in something a little bit more detailed around, like am I looking, like at the core penetration within the tumor? Am I at the margin? Am I really in the necrotin zone? Do I have, like a steric inhibition with the stromal map, like of my ADC? So that can really reveal, like the anatomical and microenvironment barrier. And so that helps us as well, like to optimize maybe, like the therapeutic concentration to help, like reach, like the target compartment. And once, like we can also look, like at the payload accumulation and as well a little bit at the toxicity. I show you, like a whole body image, like with the mass spectrometry imaging, but you can start looking, like at this payload deposition in different, like organ, and some of them, like which are the healthy organ, and definitely not, like the intent, like of the ADC deposition. So you can see, like a payload deposition in liver, on kidney, on the bone marrow. That could be, like very early sign, like of toxicity, like of your compound. You can even look, like by overlaying the payload distribution to the spatial transcriptomic capability that we can deliver as well to start, like looking at really the pathway that are responsible, like for toxicity. So you can look, like at the gene, like you are seeing, like on the right with this volcano plot. You can start looking, like at what gene, like are really upregulated in presence, like of the payload. And that could be, like pathway that are a little bit, like toxic, like ER stress or apoptosis. And that could really improve, like the early, like safety forecasting before you start, like doing your tox, like experiment in GLP manner. So we talk about, like the PK, the exposure. We talk, like about the tissue delivery, and now we're going to get into the mechanism. And this is, like, if you remember the beginning of my talk, this is really, like the third layer that's really engaged, like, and talk about, like the ADC efficacy. So we want to understand, like what the mechanism is. And the best way actually, like to understand, like the mechanism, it's really, like to look really at the target directly on the tissue section. And this is what, like our spatial proteomic, like capability are able to achieve, because we can look, like at really at the ADC, if we have, like a detection antibody against, like this drug. And we can look, like really at the co-localization of this ADC by doing, like multiple staining on the tissue section. We can look not only at the antigen, we can look at the drug, and you're going to see that we can learn much more than that. So for, like this very specific mechanistic, like target engagement in situ, this is the first layer of understanding actually, like the mechanistic aspect, like of our ADC. Our platform help us, like to really understand, like that binding of in real tissue, and that could justify, like early dose range selection, for example, and increase obviously the confidence in mechanism of action. And to go further, we can even look, like at the biological effect within, like the same tissue section. So you can look, like at your ADC, you can look, like at your antigen, can be, like a membrane receptor, but you can look as well directly at the biological effect. And you can nail down this biological effect at the level, like of the cells by staining. In addition to the ADC staining, the antigen staining, you can start, like staining your tissue section with DNA damage marker or apoptosis marker, and this is like what you have as a representation on the right like side of that slide. You can quantify like which cells are producing like those marker and help us and really like nail down like which cell are really like getting a biological effect from like ACD, and help us like to really map like what are the responsive zone like within the tissue, what are the resistant push, if I have like any other like bystander effect region. So that's threatened like really by seeing it, by looking really at the right cell where the ADC like it's really having like this effect. It's really straight on the mechanistic like PK/PD relationship. And once like you have analyzed like the biological effect, it doesn't stop like at the level like of the cells. Once your cell dies, it's going like to be releasing a lot of chemokines, cytokine. There will be cell debris, and everything like will affect like the environment actually like of your tissue. And and the ADC, like reshaping the microenvironment, it's already, like a marker of something, that could be, like a durable response in terms, like of the treatment. And here I'm showcasing an example where one of those response actually, like in the shifting of the microenvironment was to have, like a CD8. So that means, like the killer T cells, that are entering, like really the tumor, and it's going actually, like to kill it. And that's where we can define, like by looking at the CD8 infiltration, what are the responder actually, like of the ADC. It doesn't stop, like at the CD8 infiltration. You can look, like at a lot of different, like spatial features that can be, like macrophage polarization, the stromal remodeling, cytokine gradient, and all this pattern, like really correlate with durable response. So we have the capability, like at looking broadly, like to all this matter, to all, like this special feature here. And we can start identifying, like by looking very specifically at this special feature, the driver of the response of and the resistance that can guide, like just, like what could be, like the patient that's responding to your ADC, or as well, like potentially some combination therapy. And from like this immune, like microenvironment shift, we can start entering, like the prediction aspect. Now we understand, like what are the special features that could be responsible actually, like of a response. So the rest, like it's to start analyzing and correlating what is, like the special feature in the very, like baseline without any treatment, and what are the features in terms of spatial that correlated actually with that response. Could be, as I said before, just, like tumor microenvironment phenotype, the heterogeneity of the target, an immune fitness, a stromal density. And you can see, like a real example here where you have, like a responder and non-responder phenotype, and you can see, like the baseline microenvironment in terms, like of spatial phenotyping or classification of the cell. It's totally different. So we entering now, like really the hypothesis, like of finding, like the right driver for patient stratification that could help, like move, like the biomarker into the clinic. And the last slide that I wanted to showcase, it's more, like around the predictive modeling. So once we have, like that stratification, like hypothesis, we can even build a point, like more onto, like more predictivity by our ability with our AI structure here to really, like integrate the multimodality. And by this, I mean, we can start, like looking not only at the key spatial predictor that we have already isolated, but we can start implementing, like the PK data, integrated the PK data that has been provided by LCMS. We can start, like implementing some metadata around the patient. So that means that we can look, like really at understanding to a multi-layer option, like what are the responder enrichment strategy. We can support, like more biomarker informed dose optimization, and finally, at the end of the day, we can really reduce clinical development risk and attrition. And you have an example of what, like a multimodal signature look like, and you can see, like in this map from a responder to a non-responder, they have different, like expression of the different modalities. Could be, like just, like some special features, but as well, like the PK, every information that you can put in. So this is really becoming, like more precision medicine. So I'm going actually, like to end this talk today by showcasing, like that we demonstrate, that workflow, like from ADC cover, like the different, like lifetime, life cycle, actually, like of an ADC development. We talk about, like the early feasibility. We had, like some target validation. We look, like at the tissue expression. We can give you, like some clinical confidence. We have, like a good precision systemic, like bioanalysis as well, like the tissue PK. In terms, like our first in human readiness, we are already, like having, like capability for showcasing the target engagement, the PD marker where it's matter, as well, like some mechanistic, like confirmation. And finally, I just showcasing you the clinical predictivity with the response biomarker, the spatial signature and stratification hypothesis. And I promise my last slide. So what we have answer, like hopefully today, it's we wanted to give you, like a framework that help, like developer understand, like where does it go, where does it hit, where does it change, and who benefit or is it harm. And our workflow and our framework actually, like include the systemic and tissue precision bioanalysis. We're doing tissue PK, mechanistic spatial biology, and predictive modeling. And this integration, it's what makes ADC outcome more predictable. So thank you very much. And I think I'm gonna give the hand to David for the conclusion. Thank you.
All right, thank you so much, Troy and Corinne. Before we actually head to the Q&A, though, I do encourage you to please put forward your questions into the appropriate area. I did want to just give a quick plug here. Just recently, I had mentioned this about Troy, we did just recently release our results for the oligo ring trial, wherein we discussed our hybridization LCMS approaches and compared these to standard LCMS and LBA, and show how we're able to achieve much lower detection limits and higher specificity using this approach. Really removes a lot of the matrix interferences. So I would welcome everyone and encourage you, if you are working with oligonucleotides, or if you're just curious, or as Troy had mentioned, if you are maybe looking at AOCs, if you want to take a look at this release of data that we just had. So with that, I believe we can open it up for some questions. And Troy, there are a few for you already in the chat, and I think I'm going to rephrase them just a little bit. One of them was, I know during the talk you had mentioned doing kind of linker analysis, right, and utilizing whole blood. Could you maybe expand on why that would be best done in whole blood to look at linker stability rather than potentially looking at, a free payload in plasma and then doing kind of a blood to plasma partitioning calculation and trying to understand the release that way?
So obviously whole blood is a different matrix, right? You've got all of the cellular components that are going to be in circulation, and it can produce different types of results than just looking at plasma or serum based stability. And a lot of it really is, it's going to be based on the linker that you're using, what type of cellular components that may cause that degradation. And it's just something that you want to look at up front, because if you're going to be seeing it in vivo, you really want to understand the in vitro components that may lead to something that could give rise to an unexpected result in your sample analysis based on what you saw in method development and your validation.
Right. So essentially what you're saying is that whole blood better mimics the in vivo activity that's going to take place for the ADC for linkers. And that's why we want to look at it in whole blood, specifically to look at that, whether or not you're getting any type of proteolytic cleavage or otherwise, right, in whole blood. Is that correct?
Correct. Correct.
Fantastic. There's another one for you here as well, Troy. And again, you had touched on this briefly, and I think that the audience is looking to dig into this a little bit. Serum versus plasma, right? So that was briefly kind of touched upon in terms of the matrix being used. For LCMS, I believe you said that we typically use plasma as the matrix, but sometimes serum is used for LBA. Could you maybe talk about kind of what those deciding factors would be in terms of the matrix that's employed for the different analytical platforms?
So I am not an LBA expert by any means. I've never developed or kind of ran those assays. Typically, when we're doing something in serum on the LCMS side, it's usually because that sample is being used in other types of measurements, whether that's a clinical-based measurement and they don't want to collect in different tubes, and so we end up doing a serum analysis. Now, there may be something that's involved in the anticoagulating factor that may interfere in an LBA assay. I'm projecting here in terms of what might be the reason behind that. And unfortunately, once again, since I'm not an LBA expert, I can't specifically say why they would use serum in an LBA assay. Both are fine in an LCMS assay. We can use either one. We typically use plasma mainly because of any type of coagulating type factor. So the sample matrix stability is better in plasma than it is in serum.
Right. And just to add to that, Troy, a little bit, I think that you hit on this. One of the biggest differences is the removal of clotting factors when using serum. That's not usually an issue for the LCMS side because we have multiple levels of specificity. However, it can be a confounding component for certain LBA assays. I'll just add that piece in. So a couple of questions that are kind of related, so bear with me as I try to put them together for you. Kind of timelines and investment in terms of time, not necessarily money investment. So two questions. One would be, how important is it to really nail down, develop and validate a high-level method for preclinical work, right, prior to moving into the clinic? And then the second question, which is kind of I think an add-on to that, is what would we recommend doing LCMS versus LBA for preclinical species upfront work? Would we recommend going the LCMS route or developing the LBA at that point in time?
So, I mean, these are all questions, obviously, that you want to ask, and it really depends on the point of the study. If you're going to be doing a high dose tox, looking at what may be achievable with regards to an LLOQ with LCMS is definitely something that you can explore in method development, because it's always typically easier to bring a range up than it is to drive a range down. So knowing, okay, I'll be able to get to, let's say, 500 picograms in terms of the free payload. I may be around 5 nanograms in terms of the total antibody, maybe lower. It really comes down to that need. But if you know what your limitations are in early method development, but may not validate it because you know your samples are going to be very high, you don't want to dilute all of your samples into that range, you've at least made the investment, you know the potential of what you can continue to do with LCMS, and then choose an appropriate range with your preclinical. And at that stage, maybe, okay, we're not going to be able to get it with LCMS, so now let's go explore LBA if we think we're going to need those lower sensitivity limits.
Right. So Troy, if I may, let me redirect you a little bit on this. So in terms of, like, let's tie in those timelines, right? So for instance, if you have a mouse or rat, dog, whatever species you're using for your preclinical species, in terms of timelines, when we look at generating a critical reagent, the time that it takes to generate a critical reagent, and usually that time is spent generating a critical reagent that's going to be selective for human, but then you're going to be applying it for a preclinical species. What type of timelines can we provide for MDMV for, let's say, an IgG-based, IgG backbone for an ADC, if we were to look at total in that way?
So to create that critical, you're talking for an immunocapture based, it's once again going to be based on that antibody. I mean, most LCMS, we can kind of go to what's available commercially. We don't necessarily have to go into that specificity, although looking forward into human, if you are going to get into that interference, knowing that upfront and starting that development as soon as possible is always critical. But we're usually not getting into really critically based reagents for LCMS that we are for LBA, because once again, we're going to go into a digestion and look for the specificity of that peptide that we're going to be following. And it's that peptide that we're measuring rather than attaching, something on a streptavidin and then doing a second capture that may be non-specific depending on what type of interference you're going to get with an LBA assay. Right.
So I guess based on the specificity or selectivity that's conferred by the LC, by the first MS and the second MS, right? In addition to doing the pull down, what we are able to do is accelerate, right, the method development and validation timelines for preclinical species for the more standard human IgG-based backbones for ADCs. Is that an accurate statement?
That is an accurate statement.
Awesome. Apologies as I look through these. Oh, here's an interesting one as well. Is it preferable to quantify total ADC, looking at the conjugated antibody or conjugated payload? So this gets to kind of what you had outlined, looking at the different ways that you can analyze things. I guess this is really a regulations kind of perhaps question, Troy, in terms of what the agency wants to see.
Well, and it may be a feasibility question also, right? What one is going to, it's certainly going to come down to what's the regulatory agency going to want. It kind of is, half a dozen one way, six is the other in terms of what it means in terms of PK type measurement. You may be able to get to a total antibody conjugated measurement in a more relative way, I suppose. But I typically, when they come to us, we are looking at full payload versus full conjugated antibody. And that may be because that's more suitable to LCMS measurements versus LBA measurements.
All righty, thank you. Corinne, we've got a question for you now as well. So if we are looking at, for instance, a phase 1 trial, right? If the target was being expressed in, let's say, 70% of patients in the indication of interest, would you develop a selection assay prospectively to select patients, right, to put them on the trial or keep them off the trial? Or do you think it would be best to do a retrospective assessment of the target based on the phase 1 results?
So first, we will do a retrospective assessment based, on the phase 1 result. And from there, we will be able, to really nail down, what will be the real, selection biomarker that will help, moving that and validate that, for screening purpose.
Okay, thank you so much. Troy, there's a question about AOCs. So do we utilize PCR-based methodologies for oligos? I guess maybe could you go back over how we look at AOCs just briefly, and then perhaps also discuss differences between antisense oligos versus siRNA, and whether the strategies that we would employ are the same or whether they differ based on ASO versus siRNA.
So, the main difference is between an siRNA antibody conjugate is the linker part isn't going to be a part of what is in terms of the antisense strand, right? The antisense strand is going to be hybridized to what is the linker sense strand versus an antisense antibody is going to be one continuous unit. You're going to have the linker that directly goes into the antisense, which is obviously, the therapeutic part of the drug. There is going to be a difference in terms of strategy, because with an ASO-based antibody, I can directly grab onto the antibody part of it and the therapeutic portion in order to get the total based measurement, and that would be the conjugated antisense strand to the antibody. Versus an siRNA, I can't employ that exact same strategy because the free payload will have to be melted off in order to grab that antibody portion. So up front, I can do an antibody capture, right? I'll pull the antibody, which will have the conjugated antisense strand with it. And once I have pulled that out, I can then go back and hybridize to the antisense strand, and that's now going to give me the unconjugated payload. So there is a difference in strategy. The second part of that is when you get to the digestion component. So if it's not a cleavable linker, even if it is a cleavable linker, with an antisense conjugated antibody, you're typically going to get some of the linker that will go with it in that measurement. So you're not going to be able to go directly and say the antibody or the antisense strand is going to be the same in terms of the conjugated component as it is potentially in the unconjugated component. I know it's like a long-winded answer, but with the time we have, I think that's about as accurate as I can get.
Right. And I do want to also give Corinne an opportunity to address this as well, right? Looking at these in situ, right, Corinne? So how would we look at an siRNA or ASO within tissues, right? Looking in, again, ensuring that it's reaching the true site of action.
Yeah, we already have experience actually, with looking, at siRNA and ASO directly on tissue. We are using RNA-scope, technology, which is, obviously, a hybridization methodology instead, of an antibody. We are developing, probe that's going to hybridize, to our therapeutic oligonucleotide, but the same manner than for the payload, actually. So we're looking, at internalization of this ASO. We can look, at the PD effect within, the same tissue section. So, yeah, so we do have experience with it. And to look at the distribution.
All righty. Thank you for that. Because again, that brings it back, right, to reaching the true site of action, right, and our capability to provide that as well. And then there are a couple other questions here I think that I can combine for you as well, Troy, and this gets to DAR, right? So, in what situations is a total payload assay required? And, we know that sometimes there is a kind of forced degradation or forced release of the payload for total payload assay required. Could you maybe comment on those situations and when we would need to do a forced release of the payload as well as a DAR for total?
Yeah, and this comes back to, once again, the regulatory component and perhaps understanding, if you are actually seeing issues in terms of your production of your ADC. So unfortunately, we don't do DAR measurement here at Illyri. We're working on that capability, so I don't get involved in that part of the science. But what we do get involved is when we're seeing a difference in precision, say we're doing stability assessments, or we've gotten down the line and we've gotten a new lot and we're getting a different type of measurement from the same type of payload assessment. We're like, okay, we should be seeing this in terms of free payload. That's when we would be looking at a forced release in order to understand, is there a difference in what we validated in the material versus what we're seeing now, because we're getting inconsistent results.
All righty, thank you so much. So I do know that we're slightly over our time now. I would love to thank everyone for attending and for their questions today. Please do not hesitate to reach out to myself, Corinne, Troy, our team at Illyri. We would be happy to set up a meeting with you and your teams, or just have an email chat with any specific questions.
I'd like to thank you once again to our wonderful host, David, our speakers, Troy and Corinne, as well as you, our listeners, for your time and questions. You will receive an email with how to watch this webinar on demand shortly. Don't forget to visit us at bioanalysis-zone for more webinars. You will also receive an email with how to watch this webinar on demand shortly. Thank you again for attending, and goodbye for now.